Good morning and good afternoon to you all. My name is Calvin Watkins. I am a product manager at Gusmer Enterprises, and welcome to the Brewing Microbial Monitoring and ATP Testing webinar we are hosting today. I'm thrilled to announce that Jill Gallegos, our technical sales representative, will be presenting today. So I'd like you all to welcome Jill and hope you all learned something today at this webinar.
Hello, everyone, and thank you for joining. My name is Jill Gallegos. I'm a microbiologist and technical sales manager with Gusmer. I have been with the company for almost 5 years now. And prior to that, most of my experience was in microbiology and food safety. So what we're going to talk about today, we are going to talk about why we should monitor for microbes at all. Then we're going to talk about some tools we have in the lab to use. And then we're going to go to some best practices and common contamination points and troubleshooting. And finally, we will talk about ATP testing. So why do we want to monitor for microbes? So we want to ensure that there are no contaminations, and if they're in the finished product, and if there are, we would like to know what they are, what organisms they are. And then we'd like to identify some contamination points, that may be contributing to contamination in the brewery, identify equipment failures or inadequate sanitation procedures, and identify contaminated product from good product. So first I wanted to kind of tell you about some common spoilage organisms that are found in the brewery. So first we have wort spoilers, and these are aerobic guys that fall into the family of Enterobacteriaceae. Then we have lactic acid bacteria. These guys are facultative anaerobes, and basically that means when they do not have oxygen, they go into a fermentation mode of producing ATP. And example of a couple of those are lactobacillus and pediococcus. Then we have anaerobic beer spoilers, and these are pectinatus and megasphaera. Also, we have beer spoilers in the presence of oxygen, and these are acetobacter and gluconobacter. Okay, so this is a very, very detailed slide, but I wanted to include it because it's got some really great information. We will either have this webinar posted on our website or mail it out or email it to anyone who has interest, because I think this slide would be very beneficial for you guys. So basically, this is showing all the selective and non-selective media that Millipore makes and Gusmer sells to the brewing industry. So up here, you'll see the yeast and mold section. So for example, here is Brettanomyces broth. This is often an issue in breweries. So there's this media to specifically identify bread. And then there is the Wallerstein broth that will identify any yeast in the laboratory. Down here is some aerobic plate count media. This is, again, it's just a general media that will identify any aerobic bacteria in the brewery. And down here are selective medias for bacteria. So you'll see that the reason I think this is really beneficial for you guys is it has temperature and incubation times listed. It also has what the typical colony should look like. So again, this is a slide that I think is helpful. Okay, this slide is some equipment that we sell. And what I think is most important about this slide for you guys is that you are able to sample 100 mils of either wort or beer at a time. In a general micro setting without this equipment, you could only sample 1 mil at a time. And so you're getting a very large sample size. This is the pump here. This is the manifold. And these are some disposable funnels that we sell. And then this is just a dispenser for the membranes. So this is a closer up version. So you can see this is a disposable funnel. You're going to pour 100 mils of either beer or wort through the top of the funnel. And then the pump is going to pull it through. And this membrane, it's hard to see, but that's in here, is a 0.45 micron size. So you know that bacteria are 0.45 microns and yeast are 0.65 microns. So what's going to happen is all those are going to remain on top of this membrane. And then at which time you can either, with one of the other funnels, you can use a tweezer, a sterilized tweezer, to remove the membrane and place it on the media. Or in this case, you can just add the liquid media and then incubate. So I wanted to talk a little bit about the two options for media. So you have the granulated dehydrated media. And this is, going to be much less expensive, although you're going to have a trained microbiologist or someone who's highly trained in aseptic technique. You're going to have a hot plate, a flask, an autoclave, all those things in your lab to be able to make your own dehydrated media. So another option are these liquid ampules. So these are going to come ready-made, already sterilized, where you'll just add it to the Petri dish like that. So now let's talk about monitoring the brewery. So in the micro world, we talk about cleaning, sanitizing, and sterilizing. So cleaning is just the physical removal of any debris or contaminants that may be on the equipment. And then sanitation is the addition of a sanitizer to more reduce the microbial load. And then sterilization completely eliminates microbes from the process. So that's never going to happen in your brewery. So what I'm saying here is that even after doing these things, you're still going to have small amounts of contaminants remaining. And another thing I wanted to mention here on sanitation, it's beneficial for you to rotate your sanitizers. So maybe, every 3 months or so, because what happens is the bacteria become resistant to the sanitizer. So just kind of switch sanitizers every so often. And the 3 steps to brewery sanitation are clean, rinse, and sanitize. So the process sanitation. All contact surfaces must be sanitized at startup. Recommend 180 degrees Fahrenheit or more, and ensure that each surface reaches that temperature. If the line remains down for an extended period of time, it should be re-sanitized. And a good rule of thumb is if it's down for longer than 1 or 2 hours. So you think you've got your process right, you're doing everything you should, but it's important to check and make sure that the process you have set up is actually working. And that's where ATP swabs would come in. You can do temperature checks on suspect areas. You can do visual inspections of hard to clean areas. Procedure checks, make sure your operators are trained. Another tool is hotspot mapping. And the ATP system and software that Gusmer sells can really help you with this in tracking your failed spots and focus on improvements and create checklists and checkpoints during and after sanitation so that you can continually make the process a process of improvement. So these are some things that you just kind of want to watch out for during sanitation. So not opening all valves, not closing all valves while they're still under flow. This combined with drain placement is a leading cause of periodic micro hits. Not sanitizing the top of your containers like surge tanks. Failure to reach temperature due to dead legs. Outside of filling spouts. So, making sure that outside of your filling spout is clean as well. Not using adequate water flow. Typically the same flow rate the process is run at should be the CIP and sanitation flow rate as well. There should be pressure on the line. Occasionally throttle back valves to create a little back pressure for better cleaning and sanitation. So drains. These are ugly guys. Many drains have positive pressure, which can produce steam, which will push all the bacteria that's in the drain all over your equipment. Unfortunately, many lines are designed so that sampling the drain valves are located directly above the floor drains. So opening without flow or not closing under pressure during CIP will cause contamination. I've been in several manufacturing facilities where I see people pressure cleaning their floors. And so it's important to know that if you're pressure cleaning by a drain, all the bacteria that is in the drain is then being put into a mist, which will then land on your equipment. So some common contamination concerns are dead legs or long T's where the drain sample valve is, old piping, reclaim or recycle lines, filling spouts, sanitation cups allow the outer rim and portion of the filling spout to be clean, drip and drain lines, gas lines, and addition lines. Also hoses. I should have put hoses on this slide, but hoses in the brewery are definitely something you should watch out for. So biofilms, you remember when I talked about rotating the sanitizer? So this is what bacteria do in the presence of sanitizer. They will mutate over time and they kind of secrete sort of like a Vaseline, but like invisible substance over them to protect themselves from the sanitizer. And it's typically in hard to clean areas. It's difficult to remove and prevention is key. This is a good reasoning for ATP testing because biofilms are something that you cannot see. So sanitary sampling, you want to make sure you only open and close the valves while they're under pressure. You want to use the smallest valves possible. You want to spray the outlet with alcohol and allow a second or two of flow before collecting the sample. Install specifically designed sanitary sampling valves. Make sure you use sterile containers for the sample collection, and avoid dead legs between the product flow and the valve outlet, also known as long T's. So now we're going to talk about ATP theory really quick so you understand what is going on in the technology. So firefly luciferase is a light emitting enzyme that is responsible for the bioluminescence of fireflies. When ATP is combined with luciferin, light is emitted. And there is the chemical reaction there below. I included a firefly picture for you guys. So what is ATP? So ATP, or adenine triphosphate, is the primary energy carrier in all organisms. So this molecule, when found on surfaces, will combine with luciferase and produce light. ATP instrumentation that is used in food and beverage manufacturing will quantify the amount of light and yield a reading in relative light units. And here is an ATP molecule. So what can produce a positive test with my ATP instrument? Bacteria, of course, yeast, malt, hops, any material that is or was living. So why would I care if it's not yeast or bacteria on my equipment? Well, because any remaining biological material after sanitation provides the perfect environment for microorganisms to multiply. When there is a food and water source available, it is inevitable that microorganisms will thrive. So what are some good examples of ATP technology testing sites in my brewery? So the points where the product touches or product contact surfaces would be things like filler heads, fermentation and bright tanks, tank valves, and definitely transfer hoses. And some non-product contact surfaces that are good would be drains and sanitation equipment. And here's why I say sanitation equipment. So say you've got your squeegee or your mop that you're using to clean the floor of the brewery. If that mop is contaminated with a heavy load of yeast or bacteria, then you are spreading it all over the brewery. So it's important to just check that from time to time. So MVP Icon is an ATP instrument that was created by Biocontrol Systems based in Seattle, Washington. Gusmer uses the Icon system in the US under the Millipore brand. So there are several competitors that make this sort of equipment as well. But I think the thing that kind of sets ours apart from the others is that it comes with a software that allows you to create sampling plans and perform testing without writing down results. So it's very annoying to be walking through the brewery carrying a tablet, paper, the instrument, the swabs. And so this is very beneficial. And then the software stores all of the results, which will allow for tracking of failing results and therefore makes troubleshooting much easier. So Biocontrol used thousands of data points to kind of establish their pass, warm, fail results. So it makes it easier for the quality assurance tech who is sampling. to know right away. You see this large pass result. So you know right away without having to figure out the scale if the result has passed or not. So what are some important facts to remember about this ATP technology for sanitation verification? A failed result does not necessarily mean that bacteria and yeast are there, but it does mean that the surface is not clean and therefore can promote bacterial or microbial growth. It is important to test after sanitation and use the result as an indication that the equipment needs to be cleaned again before production begins. It is important to track results over time and use this information to identify trends. This technology is the quickest and easiest way to identify issues that can lead to spoiled beer before big problems arise. So now we're going to talk about the software that comes with the MVP icon. So when you receive the instrument, you will download the software and you will come over here to this section where it says raw data. So this is the area where you're going to load in your sample sites. So I know you guys like to name your tanks. You can go in here and name your tanks, put in your filler heads, and make sure those are numbered separately. Go ahead and put in your hoses, anything that you think you're going to include in your sampling plan. And then once you go out into the brewery, they are going to populate into the instrument. That way, when you're in the brewery, you can just, you don't need a paper or a pen. You can just go in and click on the sample site, take the sample, and that's it. So then when you go back into the lab and you sync to the computer, all the results are going to populate into the software. So you'll see that here. The result will be there, the time that it was taken, the date, the sample point. And then once you've had time to compile a little bit of data over time, you can use the software to do different reports. So let's take a look at the action reports. So you will see here that sample number 71 fails, the majority of the time. So this gives you the indication, okay, something is going on with sample site 71. So what you're going to do, look back and see what that represents, and that's a mixing paddle. Okay, so this tells you, okay, something's going on with that mixing paddle. You need to go and check and see, maybe it's got a biofilm, maybe it has a weld that makes it difficult to clean. So there's buildup there on the paddle. And you can do that. It's going to show you with all of your samples. When you go to home here, you can see, all the tests that failed, the retest rates, and the max data range for your certain date. So this is all included with the software and just makes the whole process much easier. Okay, now I would like to go to the demo of the device itself. This is what the MVP icon looks like. It does have a strap that you can add on to these little deals right here, so it hangs around your neck. This is what one of the individual swabs looks like. So what I've done is I've got a stainless steel surface that is dirty, so that I can demo what, a failing result will look like for you. So you just pull this out. Then you're supposed to do kind of a 4 by 4 inch square, like that. Oh, almost dropped it. Let me put that back in. Push the lever down, and you'll see that the buffer descended down here. That's to buffer out any sanitizer that may have been remaining on the surface. So these swabs only go in a certain way, so wait till it kind of clicks on the side like that. Shut it. I'm going to go in here to my test points, which were uploaded from the software into my instrument. Choose the test point, and then I'm just going to click Run Test. You'll see it just takes a few seconds. Then I remove the device. And that's where you see the failed result. Okay, so now I'm going to clean this bowl with some just spray cleaner. Hopefully I get it all off. So I've got a new swab. That in, push that down, give it a little shake. Now I'm going to push retest on the same sample point. And the significance of that is once you load that back into the software, it's going to show that this test point failed, but then was retested. So I'm running the test. And now we have a pass. Can you see that? I'm sorry for the glare. Now we have a pass. So that's how the instrument works. After you're done going around the brewery, all your sample points, you're just gonna sync the instrument back to the software, and all the data that you got for today will load into the software. So that's all for the instrument demo. If you guys want to move into any questions you might have now, I'd be happy to answer those for you.
Hi, Jill. You mentioned switching sanitizers from time to time. Does this only apply to the spray and soak sanitizers, or would you also suggest rotating the peracetic acid used for CIP with another sanitizer?
I would recommend occasionally switching up for both.
Okay. Next question. Are the pass-fail specs adjustable for relaxed or more stringent criteria? How does the instrument determine a fail?
Okay, yes, those criteria can be adjusted. As far as how does it determine a fail, going back to the sample sites that Biocontrol did on many different surfaces, including stainless steel, um, they established what would be a good rule of thumb for most environments as a pass, warn, and fail, but you are able to adjust that if you would like to based on your own environment.
Next is, there were a couple of slides we saw in the presentation that showed drains that were very dirty. Is it a common practice to clean the drains in a brewery, and do you recommend sanitizing them from time to time?
Yes, I definitely would recommend adding a sanitizer to them whenever you do sanitize the rest of your equipment. I mean, it's kind of understood that a drain is going to be dirty, but as much as you can keep that microbial load down so that you're not, again, misting it onto your equipment would be best. So yes.
One other question I see here, is there a chance I might see a clean microplate, but then see a fail on my ATP tester?
Yes, that is definitely a possibility because an ATP test is a sanitation verification tool, and therefore it tells you if your sanitation was effective. So it will pick up other things besides yeast and bacteria, like any other debris or malt, hops, that product that's on the line. And where the membrane is only going to show you yeast and bacteria.
One last question here. How important is aseptic technique for sampling?
Absolutely, very, very important. You can easily contaminate your sample with just bacteria that's on your hands or in the environment, and therefore giving you a false result.
All right. Unless there's any more questions, I don't see any more. Jill, we really appreciate your time, and thank you for the great presentation today on microbial monitoring and ATP testing.
Yes, absolutely. Thank you.
All right, everyone, have a good day. And if you have any questions, please don't hesitate to contact us at gusmerbeer.com or sales@gusmerenterprises.com, and we'll be able to answer your questions.